All right, it looks like we we have some people, so I'm going to go ahead and get started. So hello, everyone. Thank you for joining us today for the virtual Boulder Peptide Symposium. My name is Becky Knopfsinger, and I'm the chair of the Scientific Advisory Board of the Boulder Peptide Foundation. The foundation's mission is to promote research and development for peptide therapeutics. So we are a nonprofit run by volunteers, and we welcome individual and company tax donations, as well as sponsorships in order to support our mission. This year, our in-person meeting will be held in Napa Valley Marriott, September 18th through the 21st. So please save those dates and register early for the in-person meeting. The Scientific Advisory Board has worked hard to bring the latest advancements in peptide therapeutics to you for this year's live meeting, and we have many excellent speakers lined up. So please see our webpage for the most up-to-date agenda for BPS 2023 in Napa. Okay, to the fun part. Today's virtual seminar is entitled Individualized Peptides from Manufacturing to Patient Bedside. I would now like to introduce Trishul Shah. He currently is a director of business development for Polypeptide Group. He is also an active member for our Boulder Peptide Foundation Scientific Advisory Board, and he's the organizer of this virtual symposium. I'll turn it over to Trishul to introduce today's speakers. So go ahead, Trishul, take over.
Good morning, everybody, or good afternoon, depending on where you're based. Pleasure to have you all on board for this exciting seminar and presentation by both of our two outstanding speakers. So the first talk will be given by Yvonne Ware, who is an Associate Director of Manufacturing Operations at BioNTech, US, where she oversees the manufacturing of peptides and mRNA, including tech transfers and cGMP clinical trial material. Our second speaker is Kiri Potocznik, who previously led a small-scale GMP peptide manufacturing at Polypeptide, which we have called the NAP team, or the NAP lab. And NAP stands for Neoantigen Polypeptide. So during his tenure, he was also responsible for building out a dedicated GMP suite, process implementation and optimization, and day-to-day operations. So first, I'd like to introduce both of these speakers, and we'll start first with Yvonne's talk. Yvonne, I'll pass it on to you.
Great, thank you, Trishul. All right, so thanks for having me today. I'm going to talk about an overview of GMP manufacturing of individualized peptides for BNT221, which is an autologous neoantigen-specific T-cell product candidate for adoptive cell therapy of advanced or metastatic melanoma. All right. And just this is my disclosure, this work is funded by BioNTech SC, and I'm also an employee and stockholder of BioNTech. So when you think of BioNTech, if you've heard of us at all, you probably think of the COVID vaccine for Minardi, where we have a partnership with Pfizer to commercialize. But in addition to our infectious disease targets, we also have a broad pipeline in oncology, and that's where BNT221 fits under. In infectious diseases, we validated the mRNA technology. We have a flexible and adaptable platform. We've shown speed in clinical development, and we also now have a global manufacturing network. And our focus there is on significant global health needs, including COVID-19, shingles, malaria, HIV, tuberculosis, influenza, and HSV. And in oncology, we have a sophisticated toolbox of technologies across 4 drug classes. We have diverse and complementary modes of action, novel therapeutic targets, as well as potential for synergistic combinations and single agent objective responses in multiple phase 1 trials to date. Our focus there is on a broad range of solid tumors with the potential to improve treatment paradigms. So talking a little bit further about the oncology pipeline and the therapeutic modalities we have there. So we have our mRNA cancer vaccines with our iNeST and FixVac programs, where we induce multi-specificity, multivalency, high tumor antigen-specific T-cell responses with unprecedented potency. We have 4 phase 2 randomized trials there. And then in our cell therapies, which is what I'm going to talk about more today, we have our next generation CAR T-cell We have our neoantigen-based T cell therapy, which is BNT221. And we also have a personalized TCR T cell therapy. And we have 2 phase 1 first-in-human trials there. With antibodies, we have targeted cancer antibodies. We have a novel cancer cell surface targets for underserved high medical need cancers. And we have a phase 1/2 trial there. We have small molecule immunomodulators with our TLR7 agonist, which potently modulates innate immunity, and we have a phase 1 trial. Then we have our next generation immunomodulators with our bispecifics and our next generation checkpoint inhibitors to address a broad range of cancers, and we have several trials there as well. And lastly, we have our ribologicals with our ribocytokines and ribomabs, which are mRNA encoded cytokines or antibodies with potential for improved properties and half-life. And we have the potential to amplify vaccines and checkpoint inhibitors. And we have 2 phase 1 first-in-humans with the ribocytokine and 1 phase 1 with the ribomab. And altogether, we have multiple product opportunities with unique combination potential in clinical testing. So to introduce some of the immunology that I'm going to be talking more about today, we have epitopes, which in our case are peptides that get presented on MHC class 1 and MHC class 2 molecules by antigen presenting dendritic cells, which the epitopes are shown in red. And these are recognized by the T cell receptor on either CD4 positive helper T cells or CD8 positive cytotoxic T cells. And upon activation, these CD8 positive T cells release cytokines and cytolytic granules such as porphyrin, granzyme B, interferon gamma, and TNF alpha. And this induces apoptosis of the cancer cell. So in BNT221, we're leveraging this immunology. And when we talk about the epitopes, we can be talking about shared or individualized. So there could be self-antigens, which arise from expression or overexpression of normal genes, and these are typically shared between patients, and therefore the epitopes or peptides could be manufactured off the shelf and stored, depending on your shelf life, for a long time until they're needed. Whereas with neoantigens, which are antigens arising from tumor-specific mutations, in a large part, these are patient-specific and truly foreign to the immune system. So here, we're not as concerned about having a long shelf life necessarily, because these are going to be used right away with that individual patient, and we require a fast turnaround time. So this is what we're going to talk about more with BNT221. So BNT221 is a neoantigen-specific T cell product. To date, there's been many advances in cell therapies, including some successes, including clinical efficacy in a limited number of solid tumors. But there's also been several challenges which BNT221 was developed to overcome, including a single epitope approach, challenging to find potent targets. There's been toxicity observed. Starting materials are not always readily available. So with BNT221, we have some key opportunities, including multiple tumor-specific targets, which can reduce the risk of antigen escape, as well as the risk of off-target toxicity. We have an optimal T cell phenotype to drive persistence and tumor cell killing. We have broad clinical opportunity across solid tumors, as well as we have currently a non-engineered product. So the objective here is to generate a non-engineered autologous T cell product containing multiple neoantigen-specific CD4 positive and CD8 positive T cell responses that are functional, have a favorable phenotype, and can recognize and kill tumor cells. So I mentioned BNT221, where we use our NEO-STIM, which is a powerful platform to generate antigen-specific T cells, where we prime, activate, and expand these neoantigen-specific T cells ex vivo. And we have several key characteristics of this platform, including peripheral blood mononuclear cells that are used as starting material and are readily available. We raise antigen-specific CD8 positive and CD4 positive T cell populations towards multiple targets. We can induce naive T cell responses, as well as expand memory T cells, and it can be used in the context of patient-specific or shared neoantigen. The platform is also easily adaptable to allow for iterative product development, including the use of peptides or RNA as the immunogen, an opportunity for an antigen-specific cell enrichment, and also an opportunity to implement gene engineering to supercharge the products. So this is the manufacturing process for BNT221. We start off by obtaining a biopsy from the patient and sending that for sequencing, and we can get up to several hundred mutated epitopes from that, which goes into our bioinformatics pipeline, and those epitopes are assessed for manufacturability and immunogenicity. And then we get a list of 60 epitopes, which in this case are peptides, which go through peptide manufacturing. And then those peptides are pooled, and those peptide pools, along with the apheresis, are the starting material that go into our NEO-STIM cell therapy manufacturing process. And upon QC and final release, the product is infused back into the patient. In addition to this manufacturing process, we also have a characterization panel. So we look at the number and magnitude of antigen-specific responses, as well as the functionality profile of those T-cell responses, the specificity profile, and the ability to recognize autologous tumor cells. And I'll go through some of that data now. So here we're looking at the CD8 positive neoantigen T-cell populations. And you can see on the right, we have several engineering runs where we used healthy donor or melanoma patient material. And you can see that we get multiple responses per run and even up to 10 responses as shown in run 5. And on the left, we assess those responses for their functionality. And as you can see indicated by the coloring in the pie graphs, we get multiple functions from the cytokines and degranulation markers. So we have a polyfunctional profile, which has been shown to correlate with improved clinical outcome. In addition to CD8 responses, we can also look at the CD4 positive T-cell populations. So again, we get multiple responses in most of the runs, and we also see a polyfunctional response with the CD4 positive T-cells. In addition to the number and magnitude of responses from the CD8 and CD4 populations, we also look at the specificity of those responses. So you can see that the majority of the responses are highly specific for the mutant epitope, which is shown in the blue coloring. And we believe this will result in a favorable safety profile after infusion. Next, we can look at the ability of NEO-STIM to recognize autologous tumor. So on the left, we've challenged the NEO-STIM T cells with transduced tumor targets. And you can see that we upregulate CD107A and active caspase-3 in the mutant construct, which is shown in red. And this can indicate cellular killing of tumor cells. And on the right, we co-cultured the autologous tumor with the neoantigen, the NEO-STIM T cells. And you can see that we specifically upregulate interferon gamma and CD107A upon co-culturing with the T cells, which indicates that our T cells are able to respond to endogenously presented peptides. Here we have the trial design. So the trial is NTC-001. It's an open label phase 1 study of BNT221 in patients with advanced or metastatic melanoma. We look at BNT221 as a monotherapy and co-administered with anti-PD-1 checkpoint inhibitor. Part 1 of the study is for patients progressed on anti-PD-1 and received anti-CTLA-4 treatment, where we have a dose finding study and we look at 2 different doses. And then in part 2 of the study, these are for patients that are stable or asymptomatic progressor at 3 months on anti-PD-1 and anti-CTLA-4. And here we have BNT221 in combination with the anti-PD-1 checkpoint inhibitor. And this is from the dose selected from part 1, and we have up to 20 patients here. Our endpoints, our primary endpoint is safety. Our secondary endpoint is overall response rate with RECIST criteria, duration of response, progression-free survival, and immune monitoring in peripheral blood and tumor. We also have expanded clinical development in melanoma and additional solid tumor indications planned following RECIST and correlative pharmacodynamic data readouts. So coming back to the manufacturing process, I mentioned the custom epitope production, which is our peptide manufacturing, and we're going to talk about that more now. So we'll talk about the key aspects, the manufacturing process, attrition rate, and turnaround time, as well as highlight some lessons learned. So the peptide sequences themselves, I think I mentioned already, we have 60 peptides per patient. These can be anywhere from 8 to 25 amino acids per peptide. We have what we call short mers, which are usually around 8 to 12 mers, and then we have long mers, which are around 17 to 25 amino acids. These are canonical amino acids only, so we have no modifications, and these are all linear peptides. The turnaround time from when we place the order to when we ship it from the peptide manufacturing site to the cell therapy manufacturing site is around 5 weeks, and that's excluding bioburden and endotoxin release testing. And then attrition rate across several engineering runs. This is an average. So for the short mers, we have a 90% success rate, and with long mers, we have a 65% success rate, and overall that gives us 83%. So the manufacturing process, like I said, we put the manufacturing order in, then the manufacturer starts with solid phase peptide synthesis. The peptides are cleaved, precipitated, and dried, and then sent for crude and process testing. Then those peptides are purified by HPLC, lyophilized, and then the peptide powders are also sent for in-process testing. And then the peptides that meet the criteria of the IPC are pooled, and those pools are sent for release testing as well. And then the pools are stored at -80 while we review all the data and complete all the documentation for interim release. And then the peptides are shipped from the peptide manufacturing site to the drug product site. The test panels for IPC and QC testing are as follows. So we have a visual appearance, we have identity and purity by UPLC, UVMS. For the pools release testing, we also have visual appearance. And then for identity and strength, those are by UPLC, UVMS. And we have endotoxin and bioburden following the USP methods. I mentioned the average attrition rates already, but just to get a closer look at the attrition rates per run, I have that here. And overall, there's some variability from lot to lot, and this can be partly attributed to the variability in the sequences themselves from patient to patient. And on the right with the pie chart, this is a distribution of the peptides per patient on average. So you can see that we start with more short mers, but we also have a higher success rate with short mers. So we do have more short mers in the final product than long mers. And for the timeline, so this is just a generic timeline where we go from the synthesis to cleavage, purification, lyophilization, pooling, and QC. And as I mentioned, we try to keep that under 5 weeks. And then it can take several days to ship from the peptide manufacturing site to the drug product manufacturing site. And all in all, with including the shipping time, our target is 5.85 weeks, and we've been able to meet that for runs that we track the turnaround time. And then we've done lessons learned check-ins at various points in the process. So a good time to do this can be towards the end of a tech transfer prior to starting phase 1, and also towards the middle or end of a phase 1 as you're going into phase 2. So I have a few highlights from that. And we've asked the team what went well and what we can do better next time. So I've grouped those into 3 buckets. The first one is peptide selection, where we talked about there's a preference to select short mers, and that's basically due to the higher success rate that we see with short mers. We use an algorithm to filter out peptides with low manufacturability, so this helps with our attrition rates as well. And we have more flexibility to design the long mers, and I'll talk more about that shortly. It's important to have cross-functional alignment and flexibility, as well as collaboration between all the different groups, and being open to suggestions, not only from the CDMO, but the sponsor. It's helpful if they're open to some suggestions as well. And last but not least, the quality and documentation. So with the quality agreement, it's important to have detailed expectations where appropriate. And with SOPs, protocols, and methods, it's important to work together to ensure appropriate level of detail. And also with the batch records, again, working together and clear instructions for the operators and digital when possible. So I mentioned there's more flexibility to design the longmers, and want to talk a little bit more about that here. So with the shortmers, those are typically binding in the MHC class 1, and you can see in the picture in purple that it's a little bit more of a conserved binding region. So with the shortmers, the sequences that are predicted to be the epitope are the sequences that we have to manufacture, and there's not a lot of flexibility there. But with the longmers, they overhang the binding groove as shown by the MHC class 2 in red. And so these flanking regions on the N and C terminus can be modified slightly as needed to help with the manufacturability. So for example, if you had N term Q on, you could maybe add another residue or take that one off depending on where your predicted epitope is. Cross-functional collaboration. I think anybody in development or CMC is aware that this is important, but it becomes even more heightened when we're working under critical turnaround times with individualized manufacturing. So we have peptide process development and analytical development helping to review deviations and investigations, as well as master batch record revisions and providing technical support. We have manufacturing operations and sciences working on the project management and CMO communication, as well as writing and reviewing engineering protocols, reviewing and approving executed batch records, and acting as a technical liaison. Then we have our supply operations team, which manages the upstream timelines that I talked about with the patients and the biopsies and getting the sequences through our bioinformatics pipeline. They also help to ship the pools from our drug product to our drug product site, as well as to our site in Cambridge for characterization testing. And they also help with the container closure and secondary package selection. The QC team reviews all the peptide and pool QC data, as well as the C of A, and maintains the specifications and reviews stability data. And then QA oversees all of this and ensures that we're following our GMP guidelines, and they review all the same documentation, the batch records, protocols, deviations, CVAs, specifications, and investigations. So I mentioned the batch record preparation. It's important to have clear instructions in the batch records, and this can help prevent deviations and save time. There's been some instances where we implemented a decision tree as shown on the right, and these can be helpful for a situation that has known complexity and multiple paths forward. It can save time in case something happens. You don't have to get on the phone with your CDMO and your sponsor to try to figure out what to do next, and the operators can just clearly follow the instructions from the decision tree. And it's important to prepare the batch records together with the CDMO and the sponsor to ensure everyone agrees on the paths forward. Some key considerations for individualized peptide manufacturing include, first and foremost, quality and patient safety. We are following GMP guidelines for this peptide manufacturing. It's also important to have regional manufacturing if possible to help save not only on the turnaround time. Currently, our drug product manufacturing site is in the Netherlands, so it can take some time to get the peptides there. But also we want to de-risk any custom importing and exporting risks there. Instrument redundancy is important. When we're working on such a fast turnaround time, we really can't afford any downtime. So having extra additional instruments for your critical steps is important. I already mentioned collaboration and flexibility, cost effective, especially as we move towards commercialization. We want to keep the COGS down. automation and digitization can help with the COGS, but also increase the turnaround time. And sufficient capacity, so we're constantly checking in with our clinical operations team to see how many patients they're enrolling and making sure we're going to have enough peptide manufacturing capacity to handle that. So next steps for BNT221, we're going to continue with part 2 of the phase 1 trial. We're awaiting initial clinical and translational data, as well as characterizing drug product and peripheral samples to correlate with the clinical outcome and safety profile. We're opening additional clinical sites to increase enrollment rate. Right now, we have the Netherlands Cancer Institute, but we also are opening sites in Belgium and Spain. And we're hoping to expand to additional indications. And then next generation development is ongoing to improve the turnaround time and potency. With that, I just want to thank the team at BioNTech. There's a lot of people working on this or have worked on it in the past, and especially our external CDMO partners. They've been really helpful in the success of the peptide manufacturing for BNT221. So I can take any questions.
Thank you, Yvonne. For the audience, if there's any questions, you can either raise your hands and we can call on you, or you can type them in the Q&A section at the bottom of your screen. So while we wait for questions, I do have a question, Yvonne. BioNTech, of course, as you mentioned, is involved in the mRNA space. It's famous for the mRNA space, if you will. And having both platforms, how do the two work synergistically, or do they, the peptides and the mRNAs?
Yeah, at this point, for this particular program, there's still work being developed there or research. So as far as clinical trials, we're focused on peptides at the moment. But as we get more data to evaluate mRNA, that could be something we can look at as well.
Okay. Thank you. What would you say are some of the learnings that you think are key learnings that you think you've learned going through this process? Of course, the approach of immunotherapy, and I know Carrie's going to talk a little bit more about the manufacturing aspects to making the peptides, a number of peptides very quickly. What are some of the key learnings that you think BioNTech has learned from this, and that you would impart to the industry?
Yeah, so I think one thing we're looking at, 60 peptides is quite a bit. Do we need all those 60 peptides? And trying to reduce the COGS, because this whole thing is expensive. Not only do we have individualized peptide manufacturing, but then it has to go through cell therapy manufacturing. So those are a couple of things that we're trying to take a closer look at. And, we still have a little ways to go with our phase 1 trial. So we'll collect more data and analyze this further as we get more clinical data as well.
Okay. So we have
we do have somebody who raised their hands.
Okay, we actually have a question, Lauren, on the Q&A as well. So maybe we could start with those first.
Sure.
What is the regulatory status of the individual peptides? Are they GMP compliant or treated as non-GMP raw materials?
So they are GMP compliant. I know there are other trials where the peptides aren't necessarily GMP for similar applications, but I think that's more of like an academic setting. So for us, we have been manufacturing our peptide starting material under GMP.
Okay. Somebody raised their hands. Lauren, you mentioned that?
Yep. Let me, because that's actually one of the people that also put a question in the Q&A.
Okay. Since there are numerous individualized peptides for the patient, how is this taken into account during the client trial study plan?
I'm not quite sure what this is asking. I think maybe, Chad, you're talking a little bit about the number of peptides and why that number was chosen. But I think it's a starting point for us for the phase 1 trial. And as we get more data, we'll definitely take a look to see if that number of peptides is needed. Hope I answered that right.
Chad, I think you can talk. I think you're unmuted. Maybe not.
Okay. And Lauren, did you say that was somebody else that had raised their hands?
That was Chad. Yeah.
Oh, that was okay. Perfect. Okay. Are there any other questions from the audience? Great. Well, if not, thank you, Yvonne. Oh, actually, we do. The question here is: neoadjuvants are needed? Question mark. That's a question. The CD4 and CD8 cell responses do not require adjuvants.
I think there's different starting materials, different cytokines and things that go into the cell therapy product manufacturing. If you're talking about a peptide vaccine, I think they've been mostly studied in combination with adjuvants, but because this is a cell therapy, it's a little bit different.
Okay. Yvonne, one question I do have is, so your program that you talked about is sort of the T cell therapy approach. And I know that once upon a time, Neon, or what BioNTech acquired, had a inpatient program, an in vivo program. What is there an advantage of one over the other?
I think it comes down to the data and where you're going to get the efficacy. So, we're still in the middle of our clinical trial, and we'll get the data and see which one is better for the patients in the long run.
Okay. Great. Any additional questions? Okay. Well, thank you, Yvonne, very much. Very nice presentation. And I think we can then jump onto our